Hello, everyone. My name is Wahab Khan. I'm at Dartmouth Health. Thank you for the opportunity to present today at the Bionano Symposium and speak to you all about some of our work that we're doing in the chronic lymphocytic leukemia space using optical genome mapping. I have no conflicts of interest to disclose. So we can just get right into it. A little bit of a busy slide, but I thought just to kind of level set, give you a little bit of background on CLLs. It's one of the more common adult leukemias in the Western world, and one of the top recurrent leukemias in the world in general. It's characterized by this clonal expansion of mature B lymphocytes. They're noted here in the left side of the screen, and as well as CD5 positive cells that are shown on clonal expansion. There are a lot of sort of high impact findings in terms of a cytogenomics workup. There's a combination of cytogenetics and molecular that's performed that looks for well-known markers such as deletion 13q, trisomy 12, deletion 11q, 17p, and then TP53 alterations. And as well as IGHV somatic hypermutation status is also involved in this workup. And then, of course, with on the molecular end, we look for sequence alterations in various genes. Some of these are noted here: Notch1, SF3B1. Won't go through the list, but just to kind of make the point that it's sort of a collective analysis with some of these sort of standard of care assays. And then IGHV somatic hypermutation, we'll have a slide on that, and subsequently that kind of predicts disease behavior and intervention response. Current CLL FISH panels and FISH in general is a very targeted approach. Looks for a handful of these chromosomal regions that I've noted up here in this earlier bullet. And then metaphase analysis requires, in this B cell process, the addition of various adjuvants to kind of really stimulate or simulate those indolent clones to be able to get these clones to divide in culture. I'm just going to turn on the pointer here so we get some context. And so on the left side, as I noted, there's these, this dense clumped chromatin that you see in these green arrows, as well as these smear cells that are by definition much more of a fragile leukemic lymphocytes, these being characteristic of CLLs. So just to kind of give you that overview. So the rationale for us pursuing OGM in this case was to allow for the detection of these cryptic rearrangements, as well as balance rearrangements that are often not possible to pick up with current assays, and also to be able to, given the genome-wide nature of this approach, with the power of being able to pick up the structural events, to look at these, multi-hit genome profiles, and can we actually detect those? And kind of the running hypothesis was in this clinical research cohort that they may be under-detected, given how we have been ascertaining or assaying this leukemia. And all to say that, in the absence of this, the other running hypothesis was that maybe this helps to add to the relevant findings that may be incomplete, and just a broader need for more comprehensive genome-wide assessment of leukemias and lymphomas. I won't spend a lot of time on this, but just three quick things for those in the audience that are not familiar. Just to quickly note that this is a non-sequence-based approach. It uses long DNA molecules, upwards of hundreds of kilobase sized image that are imaged through these nanochannels. It's a fluorescent-based technique, so it has specific label motifs that are fluorescently tagged at these specific sites, which then creates this barcode, and then that is aligned or de novo assembled to the reference genome, and that allows us to pick up the multitude of structural variants that are noted here. Okay, so coming back to our clinical research cohort, I'll go through a little bit of our study design and then get into the results. So we did a retrospective analysis of 50 CLL samples. These were predominantly bone marrow and peripheral blood. collected in EDTA or heparin. We performed Bionano on the Sapphire system, on the Solve pipeline, and then we looked at concurrent cytogenetics with an interface FISH panel using these specific regions. IGHCCND1 was in there as well to kind of rule out mantle cell lymphomas, and then the well-known 13q, the ATM, the TP53, trisomy 12. Concurrent NGS was also performed as part of a 90+ gene panel off of an exome backbone, and then we paired this with time to first intervention data and IGHV mutation status. So we had fortunate to have sort of other data points in addition to kind of pair with our structural variant analysis with OGM. So cohort demographics, as I said, this is sort of the 50 of these that we included in this analysis. We've since then expanded on this. They were equally distributed between males and females. Median age, they were sort of the more elderly population, 63 to 65. Indications spanned, the bulk of these were CLL cases, but we did include some suspected CLL versus other MBLs or MCL type cases just to kind of give a bit more of a natural flow to these indication types as they can be presented. And so now I'll get into some of our results. So OGM detection rate and structural variant spectrum is noted in this slide. So 41 of the 50 cases had a oncogenic defined or likely oncogenic potential finding. There were about 65 of these that were oncogenic, 23 were likely oncogenic. They're sort of categorized in these on the y-axis along these SV classes, with deletions being the predominant, translocations followed by gains, complex CNV seen in about 8 cases, and then a lone duplication there. And then I'll start with the more common one. So this is well known to CLL biology, where you have this recurrent 13q loss. But what we were starting to find, and others have also noted this, is this heterogeneity that we see with these proximal 13q losses and this hotspot region on the cytoband Q14. In this one instance, where you have an absence of the retinoblastoma 1 gene, but in other cases, we have RB1 spanning deletions, and we saw both of these categories in our cohort. These were more focal and isolated and presented in more isolation kind of a overall finding, whereas the RB1 spanning were often paired with other deleterious events in the CLL cohort, suggestive of more maybe perhaps adverse outcomes. And this just shows you one such example where the RB1 spanning del13q. This also had a corresponding 11q22 ATM loss. So I've cataloged the ones that I'll show you in the subsequent slides into these categories of multi-allelic events, complex events, and then those events that were sort of OGM unique events. So I'll show you those examples. This one being these multi-allelic events tracking with a loss and a RB1 spanning deletion. This one was an interesting, we saw these rare but yet recurrent findings where we were seeing this 13q14 hotspot region being translocated to various other chromosomes. So 21q presented in this context, but also tracking with a focal deletion that spared RB1 in this case, but had this multi-allelic translocation event that was proximal to it. This has been described, these sort of recurrent 13q structural variants, such as these translocations plus deletions, have been noted to be linked to genomic instability and maybe earlier on, earlier stage CLL outcomes and shorter time to first intervention versus cases with isolated 13q, although we need a bit more data to kind of firm this up. Historically, we've seen this in the past where these, there were instances described of these 13q14 translocation breakpoints in B-cell processes involving CLLs. And then this here kind of speaks to the heterogeneity that one can see in these 13q losses in CLL, which we also saw in our cohort. So in the second category, I'll touch briefly on these complex rearrangements, structural abnormalities picked up with optical genome mapping, and they had these co-occurring copy number calls as well. So it was this overall multi-hit pattern. We saw this in about 20% of our OGM positive cases with shorter time to first intervention because we had that paired data. And we also noted the loss of TP53 being a recurring theme in these with other multi-chromosomal CMEs. And so you kind of see this almost like chromothripsis-like event on chromosome 2. The circles here are just kind of implicate the chromosomes that are involved in these rearrangements. And then just to zoom in on the copy number plots, you'll see the kind of like that chromothripsis-like profile on the 2. You'll see the loss of 8p, the 9 again showing the same events that show that oscillating pattern. And then, further down, you see some of the more segmental losses on 13q, including RB1, 17p, TP53 being impacted here, 20p loss, maybe not, maybe more of a passenger event in this instance, and a gain of 22. So going through, just the categories again, multi-allelic events, co-occurring complex events, and then now addressing some of the cryptic rearrangements, some of the more unique to OGM events. And these, in our cohort, were enriched for different types of permutations of IGH rearrangement. So in this case, with BCL2, 2 cases, and these tended to co-occur with trisomy 12s, we thought, at least we saw in our hands. We also had this very interesting double hit IGH rearrangement. There's a couple of examples I can show you in the next slide. This was sort of a case where it kind of changed the outcome of that case to a different entity, MCL, because it involved CCND1, but it involved it with the immunoglobulin light chain locus. And then we had these multi-chromosomal complex events across various chromosomes that showed both segmental losses or gains, focal losses or gains, or aneuploidies. And all these are not going to be detected by FISH. We have some folks may have an IGH sort of broader panel or reflexive panel, but by nature of the fact that it won't be in the kind of the upfront panel. And then metaphase G banding may not, of course, will not give you the gene level information, so you kind of need to follow that up. Lots of these are cryptic in nature, so important for entity reclassification, as was the case here. This just shows you this slide in picture format, where this is the double hit rearrangement in a B cell in a CLL involving proximal 2p and 14q, and then involving 19q as well as 14q. So both were, and then we had karyotype information to kind of phase these a little bit, because it noted, interestingly, both chromosome 14s were impacted. And then in this case down here, we saw a IGL-BCL2 rearrangement in a CLL co-occurring with trisomy 12. And just to kind of underscore this point about cryptic rearrangements in these complex CAVs that may not be overt, maybe more overt or covert by FISH analysis. This was a kind of a really interesting case, I thought, where we found a small 143 kb ATM disrupting structural variant that was unique to OGM in a CLL. It was an intragenic deletion with, I believe, exon, was impacting the 5' end of ATM. It's on the sense strand, and then exon 17s through 63. So it's compatible with a disruption of this, tumor suppressor that's well known to CLL biology. And then it did create a fusion transcript with a gene that was further upstream, but that was impacting the 3' end of ACTA1, creating this fusion, so the 5' to 3' fusion of ATM and ACTA1, in addition to disruption of ATM. And this was interestingly observed in an unmutated IGHV somatic hypermutation profile, kind of checking all the boxes of sort of this maybe intermediate to more adverse impact in this case. It's been described, fairly recently in this paper where, these folks found a different novel pathogenic fusion of ATM. So just kind of maybe speaking to the fact that these may be rare, but perhaps they're under ascertained because we haven't been looking for them. And was kind of nice to see that come together. Okay, and so I'll speak a little bit now to some of the IGHV data. We didn't, in the interest of time, I don't have this to show you, but just for numbers, IGHV mutated status, which is often synonymous with those events that are not as deleterious in a CLL setting. We had majority of our cases, if by OGM were enriched with either OGM negative cases or simple focal deletion 13q sparing RB1 in the majority of these cases. In other cases, we saw this unmutated somatic hypermutation pattern 43% of the time. This is frequently paired with some of the higher impact OGM profiles that I've shown you in the earlier slides. And in this third category, these are maybe somewhat of an outlier where there was this RB1 spanning deletion 13q tracking with a mutated IGHV, and perhaps this was an isolated finding, so maybe that's why we're seeing these enriched in some of these cases, suggesting that maybe that IGHV adds this independent layer beyond structural variants alone. And we paired this with our sequencing findings as well by NGS. And so we tried to then collect this data and kind of collate it in terms of time to first intervention. So we broke it up into two categories, or kind of a subcategory: high impact, moderate to low impact. This was a subset of samples, so we were limited in the number of samples on which we had this data for. So something to kind of look at larger studies from multi institutions to kind of pad these numbers a bit more to get better outcomes here. But what we found on the y-axis was, there are the months, the cases are listed here on the x, and we found that earlier intervention time to intervention were synonymous with these higher impact 28-month to intervention treatments, whereas these moderate to low impact were 71.5 months. So looking at this data a slightly different way, we did a non-parametric statistical analysis. This is the Kaplan-Meier curve, and population proportion intervention free is on the y-axis, and the time to first treatment and intervention on the x-axis. And this again shows this earlier trend towards the higher impact OGM profiles. The p-value didn't reach significance because of the number of samples we had in the cohort, but it just kind of gave this broad conclusion that, or point to note an interesting one, that early intervention in these higher impact CLL cases were tracking with the appropriate OGM defined abnormalities that were the the TP53 loss or the complex or chromoangiogenesis like patterns. Something to kind of follow up on future studies. So in summary, I will conclude by noting that that OGM revealed this multi-hit complexity and complex genome profile in CLLs, identified rare yet recurrent and perhaps under-ascertained rearrangements that were missing on standard panels, Many lesions that appeared sort of isolated by FISH were really kind of, was an eye-opener to kind of see them in these more complex genomes when assessed by OGM. So really speaking to the fact that we, if we didn't do this, we would classify that case as a different way if we didn't do the, follow-up genome mapping here. Integration with NGS and IGHV added another layer. I refined some of the important findings. In the absence of like an overt or OGM negative case, the immunogenetic profiling and the sequence level drivers are identified. So really underscoring sort of the complementary role of OGM with these other multimodal approaches that we can use or that are at our disposal. So our work here supports overarching, more precise, individualized management or potential for more individualized management in CLL, integrating OGM with these other multimodal approaches. And I want to, so I'll conclude there, and I'll just give a shout out to our team at Dartmouth and Dartmouth Health and the Dartmouth Cancer Center. This work was led by Soma Chakraborty, who's a previous HemePath fellow, currently at Yale, and then our admin team, Heather Lauren, our CGAT cytogenetics core team, Jane, Michelle, Shiva, Farzana, Kyle, Devin, Irma, our faculty, and Laura Tafe is our medical director, and be happy to take any questions from the audience. Thank you again, and I think structural variant discovery is a very important aspect of
genome biology, whether we study it in the somatic or the germline state. So I think, we have now tools that allow us to do that. So thank you for the opportunity to speak today. Thanks.